further ado, actually, I will move on to the talk. I would like to introduce Professor Corrado Poggesi, first of all, who has kindly agreed to chair. He's from University of Florence, probably one of the most beautiful cities in the world, in Italy. If you haven't been, I highly recommend you go there, but don't go in the summer because it's expensive. And Corrado, thank you again for chairing, and I leave the floor to you now.
Thank you, Davor. I am very happy to introduce this webinar from a very nice friend of me, that is Jolanda Vanderbilt, who is professor of physiology and the head of the Department of Physiology at the Amsterdam University Medical Center. She is a leader investigator in the field of cardiomyopathies. And Lili Wang, a research instructor at the Vanderbilt University Medical Center in Nashville, Tennessee. Lili got her bachelor and master degrees in China and her PhD at the Auburn University in Alabama. After graduation, she joined Dr. Nolman's lab as a postdoc, and her research interest is the study of arrhythmias mechanism in hypertrophic cardiomyopathy. So I'm very happy to introduce this webinar that is based on a recently published paper on Journal Molecular and Cellular Cardiology Plus that is based on a rare but very significant mutation in cardiac troponin T. I think Jolanda will start telling you something about this. While Jolanda is sharing the screen, I let you know that you have a special box with question and answers that you may use to ask questions during and after the seminar.
Wonderful. Thanks a lot, Corrado, for the introduction, and thanks to Davor to inviting us. It's very a big honor to be the first presenting a manuscript which is accepted in this new journal. I'm really happy that you actually mentioned that it's also very much promoting acceptance of neutral outcomes. I think that's very important, and it's good to have that task. So troponin T mutations which cause hypertrophic cardiomyopathy, and I'm very pleased to give this presentation together with Lili. I will kick off and then I'll hand over the word to Lili. Okay. Now my remote is, yeah, there we go. So I just want to first introduce the theme or where we started off, and this is a bit vague picture, but it's taken at a consortium meeting, Big Heart and FP7 funded project, which kicked off a really wonderful collaboration. And you see one of the co-authors of the paper, Charles Redwood from Oxford, our chair of today, Corrado, and I think Lucy Carrier is in the audience and will also be happy to see you all in Berlin. And another person who has been instrumental to this study is Christos Remedios, who initiated the Sydney Heart Bank, and that has been instrumental for many of our studies. Then I met Björn Nolman during one symposium which we organized in Amsterdam, and based on that he wrote a wonderful manuscript which was published in Cardiovascular Research together with Thomas Essenhagen and Christine Memory, highlighting the use of iPS cardiomyocytes, which Lili will illustrate today. And I also would like to mention our collaboration with Jose Pinto, who is instrumental in making recombinant troponin T, and he's an expert on troponin T. And it's a wonderful collaboration also there. And of course, the team of scientists who did most of the work in the lab, Vasco Segura, who's now doing a lot of experiments elsewhere, and Paul Wijnker and Lili. So basically what were the ingredients? Well, of course, international collaboration, a pioneer in biobanking, and the excellent junior scientist team, and we'll highlight innovations which we think are important in the field. So, but first to introduce the disease type we study, it's hypertrophy cardiomyopathy, and there we know that a mutation is needed to cause this disease. It results in, most of the time in expression of a mutant protein. And then what's seen in the clinic is this phenotype with thickening of the septum and often obstruction of outflow of blood to the aorta. And there's dysfunction seen, mostly impaired relaxation of the heart and actually an increased ejection fraction. And the main question is like, okay, you start off with this genetic change, the mutation, which in most people is heterozygous. So only one allele is affected. But we cannot currently prevent onset of the dysfunction and the remodeling. So we still want to learn more about what changes these mutations trigger and how they end up in cardiac dysfunction. Another thing which is really notable is that there's a large clinical variability, both at the level of the age of onset. So some people develop severe disease at the age of between 15 and 20 years of age, and others might have mild symptoms at the age of 80. So also the disease severity is highly diverse, like ranging from really severe heart failure, in some cases acute cardiac arrest, but also people with a very mild cardiac phenotype. So that has been a challenge. Now we know that based on the genetic studies that approximately 50% of all patients are identified to have a pathogenic gene variant, so a gene mutation, and that it encodes most of the times for one of the sarcomere proteins. And the most frequently affected genes are myosin heavy chain, so the thick filament, part of the thick filament, myosin binding protein C, and in the thin filament, it's troponin T, and that's the topic of the manuscript we recently published. So what have been done in the past, and what have we studied in the current study? In collaboration with Jose Pinto, we were able to use different, or to study different mutations in the troponin gene at different locations. And the K280N is in the C-terminus, but one of the questions we addressed in a previous study is mutation location. Is it how is it changing cardiomyocyte function? And the way you can do that is use so-called exchange experiments. So basically, you can make two different mutant proteins, recombinant proteins, which carry a specific troponin T mutation, and then by using human cardiac tissue samples, you can do an exchange experiment. So basically you isolate single cardiac muscle cells, so you end up with sarcomeres. And in this case, you could, for instance, use non-failing donor heart cells, given high dose of recombinant mutant protein in the incubation medium, and there will be competition, and the mutant protein will exchange or basically kick out the healthy protein, and you end up with a sarcomere which contains both the healthy and the mutant protein. So it's a wonderful way to understand how a mutant protein, in this case in troponin T alters myofilament function using human cardiac tissue samples. Now, what we've seen in a previous study, also published last year in the journal in JMCC, is that depending on the location of the mutation, and I showed you those in the previous slide, the change in function differs. So and we know that mutations which are related to hypertrophic cardiomyopathy increase calcium sensitivity. So at a lower or a specific calcium concentration, you end up with a higher force development with a mutant troponin T protein. But this shift, the magnitude, depends on the mutation. And this has also been beautifully shown in mouse studies, actually, from the team of Björn Nolman. And basically here we show it if you use a human system, that indeed a specific mutation has a different effect on the calcium sensitivity. And I also would like to highlight then that using mouse models with different mutations in troponin T, and this is the team from Florence, from Corrado Poggesi in collaboration with Jill Tardif. Basically what you see that these different mutations trigger different cellular changes. So, and that's of course an important step to make. You have a mutation at the sarcomere level, but you really end up with different changes in calcium handling mechanisms and remodeling. And that's sort of the starting point for the current paper, because here we want to link the changes with the sarcomeres at the sarcomere level introduced by the mutation, which we can study using human cardiac tissue, which we obtained from the patients, and also using this exchange method. But we of course want to understand how this also alters calcium handling of muscle cells. And that's where we come to the iPS-derived cardiomyocytes. So that's the step we aim to do in the current study. So to go to the results of the paper, what's really intriguing and what I really want to emphasize, the number of cardiac muscle samples from hypertrophic cardiomyopathy is actually NS1. So it's one heart, which was for our studies unique. And why is it unique? And there's a lot of text on this slide, but it's, I think, important to introduce it. But this patient carries a homozygous mutation in troponin T, and that's unique because most of the patients have a heterozygous mutation, and having a homozygous mutation means that there's 100% of mutant troponin T protein in the cardiac muscle cells. And it gives us a tool to use this unique sample to study the effect of these specific mutations at different expression levels. And I'll come back to that later. So we obtained this heart via the collaboration with Christos Remedios and used the explanted heart. It was a male who underwent cardiac surgery at the age of 26. Another thing is that we could compare the free left ventricular wall tissue with the interventricular septum. And we know that the septum is most severely affected. So this explanted heart allowed us to do a comparison. There's hypertrophy of both septum and the free left ventricular wall. And there are the typical characteristics seen for hypertrophic cardiomyopathy, so fiber disarray, and there was a moderate degree of interstitial fibrosis. And we had a larger group of non-failing left ventricular cardiac tissue samples for comparison, 7 males and 1 female, and slightly older. So the first step, just to highlight, we use this sample to do exchange experiments. And that's why this homozygous characteristic of the heart is so unique. So basically we here do the reverse experiments. we put in this case, healthy recombinant protein and replace the mutant protein by a healthy protein. And then you can vary the amount of mutant protein. And I'll come back to that during the experiments or the results. So step one was to characterize this human cardiac tissue. And the first observation when isolating single cardiac muscle cells and activate the myofilaments with calcium. So we basically remove all the membranes with Triton, and we can study the sarcomere properties. We observed that the maximal isometric force development is significantly lower in these cells, both from the interventricular septum and the left ventricular wall compared to non-valent donor samples. And what we conclude based on previous studies is that this is most likely due to a reduction in the myofibril density, because there's hypertrophy of the ventricular wall. We know that there's hypertrophy of the cardiomyocytes, and we've observed in previous studies that there's a reduction of the myofibril cellular density. So it's most likely not linked to the mutation directly, but mostly to the secondary, it's a secondary disease remodeling effect. Then, in line with previous studies in human samples, but also in cells from animals, we observed an increase in calcium sensitivity. And this increase or reduction in EC50 is pretty similar in cells from the interventricular septum and the left ventricular wall. So then the question comes like, is this increase in calcium sensitivity mutation related, or may it be related to post-translational modifications? Many studies have shown that in particular phosphorylation of these sarcomere proteins alter calcium sensitivity. So we moved on, and the first proteins we really focus on are the ones which are phosphorylated by protein kinase A, because that's activated upon beta adrenergic stimulation. And quite surprisingly, actually, we did not find a difference in phosphorylation of myosin binding protein C. So this shows the whole expression pattern of all contractile proteins. And this is ProQ diamond stain to stain the phosphorylated proteins. And it's surprising because in all other previous studies we've performed on cardiomyopathy samples, which are taken during myectomy surgery, we actually find a lower phosphorylation of myosin binding protein C. And the troponin I phosphorylation is only lower in the free left ventricular wall, but not altered in the septum. So this was quite opposite to previous observations in myectomy samples. Then, of course, we went on and also want to know if the phosphorylation of the mutant protein is altered. And actually, we do find a low troponin T phosphorylation. And that's also, we've studied many human cardiac samples, and we actually never found a sample where phosphorylation of troponin T is reduced. But in this sample with a homozygous troponin T mutation, there's a significantly lower phosphorylation of troponin T. And we also observe a higher phosphorylation of myosin light chain 2. And this is another way to look at the phosphorylation changes to confirm if what we see on the ProQ diamond stain is indeed true. So it's sort of like using another method to confirm a 2D gel to show that. So the phosphorylated proteins are more at the acidic side. And if you zoom in, here is troponin T. This is the septum sample, the free left ventricular wall, the non-valent donor sample. This is the troponin T pattern if you zoom in. In the non-valent donors, we know there's approximately 70% of the total troponin T is phosphorylated, but you basically shift to 70% unphosphorylated troponin if you go to this mutant protein sample. And myosin light chain 2 also here, you see the donor samples, mostly the unphosphorylated forms to isoforms, while this is shifted to higher phosphorylation in both the septum sample and the left ventricular wall sample. So there is a highly changed phosphorylation background, and the first way to try to link the mutation with the functional changes, or address if it's rather the post-translational modifications which cause this increase in calcium sensitivity, is use kinase and phosphatases to sort of normalize the phosphorylation background. And the first one we did is like an incubation with exogenous protein kinase A to have the same level of phosphorylation in your mutant sample as compared to your donor sample. And after the protein kinase A incubation, you basically see that the difference in calcium sensitivity is not taken away. So there's still a high calcium sensitivity in the cells which carry the homozygous mutant troponin T. We did the reverse, so we used alkaline phosphatase to dephosphorylate, in particular troponin T. So you can see that here. This is the non-treated sample from the non-failing. If you incubate with alkaline phosphatase, you significantly reduce your troponin T phosphorylation. And so this results actually into an increase further. It shifts both curves to a higher calcium sensitivity, but it does not take away the difference in calcium sensitivity between the mutant cells and the non-failing cells. So it appears that there's still this mutant-mediated increase in calcium sensitivity. Now, a second step to establish the mutant effect and also look at the level of mutant protein that is basically needed to increase calcium sensitivity is this exchange method, where we can introduce the healthy protein into the mutant cells, and we can either introduce a low level of mutant protein or healthy protein, or a higher dose of mutant healthy protein, varying the concentration of the mutant protein, basically. Now, this is one of the key figures in the manuscript, but it's a lot of information, so I sort of took it apart. And firstly, so we incubated the isolated cells from the mutant sample, which expresses the mutant troponin with different levels of healthy wild-type troponin T with a MIGDAC label. So you can see on the Western blot that if you increase the amount of recombinant protein in your solution, there's a higher exchange. So you have more wild-type protein in your muscle cells. And what I note here is, please note that this is non-phosphorylated troponin. So you introduce non-phosphorylated troponin into your mutant muscle cells. And then what we see is that using this exchange and lowering your troponin T mutant expression level to 14%, we basically increase calcium sensitivity. We lower EC50, which is sort of counterintuitive because the mutant protein, we believe, is also increasing, but it's the reverse, right? So you have a lower level of mutant protein and you actually increase calcium sensitivity. But please note that phosphorylation of your pKa sites on troponin is important to lower your calcium sensitivity. We actually introduce non-phosphorylated troponin into the system, and this would increase calcium sensitivity. So we believe that this change, this increase in calcium sensitivity or a lower EC50, is related to introducing non-phosphorylated troponin into your mutant cells. So what we also did is do the opposite. We used non-failing donor cells and basically introduce mutant protein into the healthy cells. And here we see, again, you introduce non-phosphorylated troponin into your system. So there's an increase in calcium sensitivity reflected by a lower EC50. So what does this tell you about the mutant effect? Well, basically, we think this reflects the effect of basically the pKa-mediated phosphorylation of troponin I is known to lower calcium sensitivity. So if you take that away, you increase calcium sensitivity. So we performed all these measurements again, but then after incubation with protein kinase A to normalize, to maximize the phosphorylation background of troponin I. And then what you see is that actually the response to pKa, so if you measure before the calcium sensitivity before and after treatment with protein kinase A, is increasing depending on the amount of mutant or unphosphorylated troponin that you introduce into your system. So for instance, here you have 86% unphosphorylated troponin into your mutant cells. So the pKa response is largest, and that's what you would expect. And the same is seen for the non-failing cells. If you introduce, if you take away sort of 50% of the healthy protein and introduce the mutant protein, which is not phosphorylated, they respond to a large extent to protein kinase A. And then we look at the level of calcium sensitivity, which we believe reflects the mutant effect. And then we see that indeed the calcium sensitivity is still higher compared to non-failing donor cells. And also if you compare them to donor cells where we did an exchange with wild-type protein, having a similar phosphorylation background of troponin I by pKa sort of uncovers the mutation effect. And what's striking is that this shows that already a very low level of mutant protein is able to increase calcium sensitivity, and it's not increased to a larger extent if you increase the level of mutant protein. And that basically comes to the first or the conclusion of the studies we've performed using these human samples, is that a low expression of this K280N troponin T mutation is already sufficient to increase the basal myofilament activation in human HCM. But of course, this was part one, using the human cardiac tissues and looking at the myofilaments, but we also wanted to know how this mutation would alter properties of intact cardiomyocytes. And therefore, I'm happy to give the word to Lili, because she can wonderfully explain how those experiments were done.
Thanks, Jolanda. So before my talk, I just want to see, like, as we know, a lot of the HCM study, the hypertrophic cardiomyopathy study, is done in the mice. But as we know, mice is so different than the human. For example, mouse heart are much smaller and a bit faster. Because mouse heart, this is because mouse heart has a different like the ion channel expression, especially the most important replication. Sorry, I cannot control these slides.
You want me to do it? Otherwise,
yes,
Lili?
Yes, please. Thank you.
Yeah.
Yeah, so especially the IKR and IKS did not express in the mice, but like abundantly expressed in the human cardiomyocyte. Can you please, Jolanda? Okay, yeah. So the mice just have the very major limitations as a model of the human cardiac disease. Unfortunately, human tissues are really hard to get. So our question is, how can we model the human cardiomyocyte? So the techniques of the human-induced the pluripotent stem cell iPS cells just can help us model the human cardiomyocyte in vitro. So what is iPS cells? So we use a skin biopsy to get the skin fibroblast from the healthy volunteer, and reprogram this fibroblast into iPS cells. And these iPS cells can differentiate to the different type of the cells. So today I will focus on the cardiomyocyte. So in this study, we use a small molecule method to do the cardiac differentiation. Basically, we use a chair to drive iPS cells into the mesoderm cells, and use IWR to drive mesoderm cells specific into the cardiomyocyte, and culture these cells for 30 days. As we know, this day 30 cardiomyocyte expressed like the cardiac specific markers like the sarcomere alpha actinylate, myofilament proteins. And also this iPS myocyte have the calcium expressed the calcium handling related proteins. But these iPS cells have the different morphology and very weak contractilities. So how can we improve that? So this is one example just to show like if we culture the iPS myocyte just on the 1/60 diluted matrigel, this is really hard kind of the substrate. You can see the different type morphology of the myocyte, and the sarcomere have the disorganizations, and this myocyte has really weak contractility. But if we plate this myocyte on the Matrigel mattress, this Matrigel mattress actually provides a physiological load to this myocyte. We cultured these cells on this flexible substrate for 3 to 5 days. We can see a lot of the rod-shaped cells and with organized sarcomeres. More importantly, the Matrigel mattress have very strong contractilities. So back to these mutations. So as I mentioned before, we generate the control iPS cells from the healthy volunteers. And now we use CRISPR-Cas9 to knock in this mutation to the control iPS cells. And we got one heterozygous and one homozygous, the iPS cells. And we use the cardiac differentiation to differentiate these mutant cells to the myocyte and do the phenotyping. So for this study, we ask 2 questions. Firstly, this mutant iPS cardiomyocyte can recapture the clinical phenotype of the HCM in the dish. As Jolanda mentioned before, this mutation is increased calcium sensitivities. So our next question is, does this mutation affect the intracellular calcium handling in this myocyte? So to answer this question, we plate control and mutant iPS myocyte on the Matrigel mattress for 3 to 5 days. And we measure the cell shortening by the age detection and measure the calcium present by the fluorescence dye Führer 2. So contractility, what we find is, in the resting stage, just a diastonic phase, the cell length did not change between control and mutant cells. But at 0.2 Hz, the cell and 0.2 Hz pacing, the cell shortening significantly increased in the heterozygous and homozygous mutant cells. Time to peak for the contraction did not change, but homozygous mutant cells have the prolonged time to basal 90%. That means like the homozygous mutation just impaired the relaxation. So based on this data, we know this mutant iPS myocyte just captured the key disease features of the HCM in the dish. So nextly, we look at the calcium. And the diastonic phase, because like the increase of myofilament calcium sensitivities, that means calcium of the myofilament is much slower. So as we expected, the diastonic calcium level significantly increased in the heterozygous and homozygous mutant cells, but did not change the calcium amplitude, calcium present amplitude. Did not change the time to peak. Significantly prolonged the calcium present time to basal noise, and increased the calcium decay, slowed the calcium decay. So based on our study, we know that the TNT K280N, this mutation, increase the myofilament calcium sensitivities and increase the diastonic calcium levels, slow the calcium decay, and cause the hypercontractility and impair the relaxations. So we think this IPS model can perfectly model the human HCM in the dish. So this model can be for future disease kind of the studies and also for the drug discovery. Thank you, Jolanda.
Thanks a lot. This sort of summarizes all the observations in a graphical abstract. And I think another thing I would like to highlight at the end of this presentation is that I hope we convince you that it's good to invest in biobanking of tissues, but also stem cells, so blood samples to put in a lot of efforts to make iPS-derived cardiomyocytes. And I think what's really worth is to compare iPS-derived cardiomyocytes with characteristics which we see in the human phenotype. And I would like to point out also a recent paper from which we wrote with 2 working groups of the European Society of Cardiology. And basically the team led by Thomas Eschenhagen put forward some recommendations of where do we still need to invest, pay attention. So one is, of course, reduce experimental variation. So really work on standardized protocols, improve maturity. That's a very obvious one maybe, but there's still a long way to go. And I think it's good to, we hope with this, the discussion of this paper, we start discussions, and I think we should make it an ongoing discussion to improve our systems, really improve our functional readouts. It's good to characterize them very broadly. And also what's worth is to stress these iPS cardiomyocytes and see if that provokes disease, because I think what is really striking is that we observe already changes in the heterozygous iPS-derived cardiomyocytes, and that's really important because often using, for instance, animal models, the heterozygous do not show at least not a disease phenotype if at a young age. So I hereby would like to thank you also on behalf of Lili for listening to us. And I hope we can discuss challenges and solutions for these models, and we are happy to answer your questions.
Thank you very much, Lili and Jolanda, for this very nice presentation. And according to the questions and answers, there is a question by Daniel Johnson. You can read the question. Very interesting data. Thank you. I had a number of questions. First of all, did you manage to look in the atria of the human sample, or did you try and produce IPS with an atrial phenotype? Given that atrial fibrillation can occur in, I believe, over 20% of HCM patients. Secondly, given the pKa phosphorylation, did you challenge the iPSC with beta-adrenergic stimulation or look at potential changes in PDE compartmentalization?
Shall I address the first question? No, it's interesting, and I think there's little information about the atrial phenotype. The Florence group has some really nice data there. In this case, we didn't study the atria, but I think it's still, there's a lot of work to be done still. That's maybe you can answer it better, Corrado, what you observed in your studies, looking at the atria.
I think it's a very interesting point, and actually I had talked with Christos Remedios when he came to Florence a couple of years ago, before the pandemic, and I was asking about atrial samples from this same patient, and we should have written a project to pursue this. And we are still thinking about that, but I'm sure that Cecilia, for instance, Cecilia Ferrantini will be very eager to go on with that.
Yeah, no, I think because it really adds to the clinical phenotype, right? It's really a sort of a vicious cycle, but of course the mutation may itself already introduce cellular changes in the atria. Yeah.
All the questions raised are very interesting. Just the one also on the IPS. Maybe Lili, you could say something about this.
Yeah, that's really interesting question. We did not try this IPS cells like for the HR differentiations. So mostly we want to see like what happened to ventricular myocyte. If we see the phenotype, we can continue study on the atria. And also for this IPS ISO challenge, we did not do this like on this study. So we want just the first lazy and without any challenge, what happened to this mutation and the basal 9, either they affect the contractility or they affect the intracellular calcium handlings.
Okay. There was a question by Christos Remedios asking how many examples of homozygous genes are known.
I think we know of this is a rare one because it's also a rather high age, this person, because the ones I know of are pediatric cases, and we know of some children who end up with a homozygous mutation in myosin binding protein C, for instance, or a compound heterozygous mutation, so they are unlucky, end up with a mutation in myosin heavy chain, and for instance also troponin T or myosin binding protein C. But those children do not become old, so they have a very severe cardiac phenotype after birth. So that's the one I know of. But Chris, it's really nice that you're here, that we don't see you. That's a pity, but I think this is really one of the rare human samples I know of having a homozygous mutation.
There was a question by Moni Nader, not on the answering question block, but in the chat. Interesting talk. Have you tried to challenge these cells with adrenergic agonists and check their contractility? Lili? It's in the chat.
Oh, I see. No, we did not. As I said before, so first thing is like we want to see like what happened to the basal 9. So we did not do any challenge to the cells, but we will do this later. That's really interesting.
Thank you. And there is a question by Lucie Carrier. Hello, Lucie. How are you doing? She thanks for the presentation, and she asked a couple of questions. One on IPS lines. It looks like you also obtained on target effect of case 9 in your heterozygous line supplemental file. How much did you check in deeply? This is the first question.
Oh, should I come? Should
yeah.
I? So yeah. So for this heterozygous 9, like we for the heterozygous line, do you mean like how we check this is really heterozygous mutation? So for this one, it's like we usually do CRISPR and do first colony screening. And once we get the heterozygous line, and we will do the second screening, pick the subcolonies from the heterozygous colony to further confirm all the colonies just carry the heterozygous mutations. So for this line, we did this way to further confirm this is really heterozygous, not like a homozygous mutation mixed with a wild type.
The second question by Lucy was biobanking for iPSC. How do you want to do it at a European international level? Master cell bank, or what about the soap?
Yeah.
I don't know who would like to reply.
I would love to address that. I think it's a very relevant question, and so I think there are several items which at least I find important. Is that the collection of the blood samples should be from patients which are carefully characterized. So I think we also need to have sort of a minimal data set of clinical characteristics linked with these lines. So that's, I think, step one, which is super important. Then, of course, it's important to have genotyping so that we know the actual causal gene. And then, the standard operation protocol is key. I agree, it would be really important to get a consensus of how different labs at an international level would collect store the blood samples and subsequently do the whole differentiation. I think it's important, but I think looking back at what at least we just started this up in Amsterdam, so we are really new to the field, but I think more and more people are now set up to move on and to start doing functional studies. So I think it's really a nice time to team up and to jointly think of a good strategy. That's and convince policymakers to of course, and foundations to invest heavily, because that's a major hurdle. I hope I addressed this.
Yeah, I think so.
Yeah.
We have a question from Mike Regnier. Could you comment on whether the critical mutant content to affect function may be dependent on where the mutation is within a sarcomere protein, or which protein the mutation is in? Jolanda.
Yeah, so, or which protein the I think, so what we've now seen for, I have to read it on where the mutation, so we, based on the 2 studies we've now performed on troponin T mutations, we see that mutation location really defines the degree of change in calcium sensitivity. So I think I'm pretty convinced that we now have solid data, and also if you compare it to the studies done by Björn Nolman and by Corrado's group and Jill Tardif. So for troponin T, I think that's really mutation location is super relevant. But then there's an additional level of complexity, because we also know that mutations in myosin binding protein C are often truncating mutations, not causing a mutant protein, but rather lower level of mutant protein. So that's an entirely different disease mechanism. Yeah.
Okay.
Corrado, any reflections on?
No, there is an additional question from Alexei Vornikov, and the question is related to the basal level of cTnT phosphorylation. Is there a crosstalk between mutation and phosphorylation site? Did you do in vitro pKa treatment assay in the mutant TNT?
Yeah, that's a good question. We checked it. So PKA was able to phosphorylate the troponin T actually. So yeah, that's. But I didn't include it in the present presentation. Yeah.
Okay. I also had a question, probably for Jolanda. It's a kind of interesting question. Did you see any change in the resting tension?
That's a good question, and I don't think we did actually. That's if any. No, we didn't. But I must say, for this preparation, I'm not entirely sure if we corrected it already for myofilament density, because that's important to do. So
yeah,
I must
because the tension
say,
is affected by the density.
yeah, and you do the beautiful isolate myofibrils, so you would know better. You did find higher passive force, right?
Yes, I think we had the data about the resting tension that at least the suggestion was that we were unable to restore the normal resting tension while replacing the mutant troponin with the wild type troponin, because we were unable to exchange 100% of the protein, but 75%. So
yeah.
That may be a kind of consistent data with yours. I mean, that it was enough, a small amount of protein to produce an increase in resting tension.
Yeah,
it could be a nice thing to check. And I had a last curiosity. I'm a bit ignorant, but I know that troponin T have several isoforms due to alternate splicing. Is the mutation present in all the isoforms?
Lili, do you know that? Good.
I honestly, I don't know. But I know like the IPS myocyte definitely have the different troponin T isoforms. As Joseph Pinto, he mentioned that before, is like the fetal isoform most present in the IPS cardiomyocyte.
Yeah, I asked because in the IPS you have a kind of very early representation of the cardiomyocytes. No, in any case, thank you very much. I think it was a great talk and great information, very nice. And I'm sure that we will ask Christos Remedios the samples from the left atrium.
He found atria. He dove into the. It's good. Yeah. Well, thanks to everyone who is not visible and actively participated.
Lucy, okay. I won't see you in Berlin, Lucy, and I'm sorry about that, but I was just back from the States and I'm not allowed to go to. No, I'm kidding.
Well,
so Davor, I think
I mean, first of all, I'd like to thank all of you again. Really, really wonderful presentations, interesting work, and it's Corrado. Thank you for all your knowledge and wonderful experiment, unbelievably effective chairing as well. And thank you to the audience and lots of questions, lively discussion, and see you all in Berlin, hopefully. And for those of you that are not there, hopefully see you soon in person. And we'll stop here. And if you want to watch it again, it's going to be on YouTube in the next 24 hours, I hope.
Thank you.
Thanks a lot.
Bye, everyone. Bye. Thank you.
Thank you. Goodbye.
Thank you.
Ciao.